Hello and welcome back to another video. In this video, we're going to solve Biology Paper 5, October-November 2024. Let's begin. Okay, so this is question number 1. Let's read question number, the first question, and then we'll see what is it that we need to do. Let's start reading and understanding first. Okay, so it says, algae are a diverse group of photosynthetic eukaryotes in the kingdom Protoctista. What are eukaryotes? Eukaryotes are cells which do have a nucleus inside them. For example, plants, animals, fungi. On the other hand, bacteria is a prokaryote. It does not have a nucleus. It only has a DNA. Then these are two species of algae that live in seawater. Figure 1.1 shows a scanning electron micrograph of R. salina, which is one of these species of algae, which is a unicellular organism. R. salina is red in color. Figure 1.2. The second figure shows the electron micrograph of the second species of algae, which is also a unicellular organism. The individual cells can grow together in a chain. And you can see this is one cell. This is one cell, and then these are multiple cells arranged together in a chain. This is yellow-brown in color, while the first one is red in color. A student prepared extracts of the photosynthetic pigments from R. salina and S. costatum. So from the two species, the student extracted the photosynthetic pigments. And then he carried out chromatography to identify and compare the pigments in the two species. These are shown in the figure below. Okay, so we have two chromatograms with pigments extracted from both species. The question says, use figure to calculate the RF value of alloxanthin, this one from R. salina. Remember, R. salina is here, mentioned there. Now, remember the formula for RF value. This is the distance traveled by the extract or the distance traveled by the solute divided by the distance traveled by the solvent. So what is the distance traveled by the solute? This is going to be from the origin all the way to the center of alloxanthin. So over there, okay? It's going to be this much. This one. Divided by the solvent, it's going to be starting from the origin all the way up to the solvent front. You're supposed to divide them together and you will get 0.4. Remember, it has no units. Okay. Table 1.1, this table here, shows RF values of some photosynthetic pigments. So the RF values are given as well as the name of the pigments. R. salina, the first one, lacks two photosynthetic pigments that are present in S. costatum. Okay, so two of the pigments are lacking in this one, and they are present in the second species. Use the figure and table to identify the two pigments that are present only in the second one. Okay, so we have to find out which of these pigments are present in the second one but not in the first one. So you have to compare the lengths or the distance traveled by both of the solutes, by all the solutes. Okay, so if you see this one and this one, they are the exact same height, so they're both same. If you look at the last one, these are all this one has also traveled equal distance from the origin, both of them, so these are also identical. That means this pigment and this pigment is absent in R. salina. So you're just supposed to find the RF value of these two pigments. Again, that this is going to be the distance traveled by the pigment or the solute divided by this distance from the origin to the solvent front. Okay, and when you calculate it, you will know the values are 0.43, this one, and 0.32. Okay, moving on. The extract of photosynthetic pigments from each of the two species was used to obtain absorption spectra as shown in figure 1.4. Okay, so on the x-axis we have the wavelength, and on the y-axis we have the absorbance. It shows how much of a particular wavelength of light is absorbed by each of these species. The student planned to investigate the effect of light wavelength on the rate of photosynthesis in R. salina and R. costatum. The student planned to determine the rate of photosynthesis in two species when they were exposed to three different colors of light. Blue light with peak wavelength of 455 nanometer, somewhere around there. Green light, peak wavelength of 550 nanometer. And red light, peak wavelength of 670 nanometers. Okay, use the figure to compare the expected rate of photosynthesis for the two species in blue, green, and red light. Okay, let's mark where is blue light, where is each of these lights. So 455 is blue light. This one is 455, which is blue. Then we have green light at 550, so this one is green. And then red light at a peak wavelength of 670 nanometers. So this is going to be 670 nanometers, and that is red. So you have to see what is the rate of photosynthesis. How can we relate the rate of photosynthesis with the wavelength or absorbance? So we can say that if there is a greater absorbance, the rate of photosynthesis would be higher, because remember, photosynthesis, for photosynthesis to occur, we need light, right? Light energy is required. So if there is a greater amount of light energy that is absorbed by the leaf or by the pigment, the photosynthesis would be higher. So if you look at blue light, 455, this is at this point. So you can see that our S colastum, this one, will have a higher amount of photosynthesis because there is greater absorbance. Now let's look at green light. So if you see, green light is here and at 550 nanometer. So this one, you can see that there is a slight difference, but S costatum still has a higher rate of photosynthesis. We'll have slightly higher or slightly greater rate of photosynthesis. Now let's look at red light. So at red light, which is 670 nanometers, you can say that they're both exactly equal. The rate of photosynthesis in both of the species will be same. Okay, moving on. In the investigation, the student used three different filters, blue, green, and red, to expose the two species to different colors of light. A blue light filter would only allow blue light to pass through while preventing the other colors to pass through it. This is the same for green and red filter as well. So he or she uses three different filters to expose the two species to three different colors of light, blue, green, and red. The student immobilized the algae in sodium alginate to form two sets of algal beads. Okay, so one set for R. salina and one set for R. costatum. So you can see these are algal beads. They all have the same size. Photosynthesis of the algae is not affected when they're immobilized in the beads. So the rate of photosynthesis will remain same. The student placed these algal beads in small bottle. So this is a small bottle as mentioned here in the experiment, and it also has hydrogen carbonate indicator solution. This is used to estimate the rate of photosynthesis when the algal beads were exposed to each color of light. The indicator solution changes with color as shown in table 1.2. Okay, so what happens here is that we have a filter and light is allowed to pass through. The student is examining when the color of light changes, how does the rate of photosynthesis changes. When photosynthesis occurs, that means the algal bead takes in carbon dioxide and produces oxygen. There's going to be a change in acidity. Hydrogen carbonate indicator is going to change its color with the changes in acidity. Remember, carbon dioxide is an acidic gas. When carbon dioxide is reducing, the acidity is reducing. So let's look at this. So you have the color of indicator solution. The color can change through this range. This is the pH. When carbon dioxide is increasing in concentration, it's becoming more and more acidic, the pH is reducing. At normal atmospheric concentration, it's 0.04% and the color is red. And then as the carbon dioxide is decreasing, that means the photosynthesis is being occurred because it is taking in carbon dioxide. The pH is going to increase. So the color will change from red to purple. The rate of photosynthesis is increasing as carbon dioxide is reducing. So the question is, the student used pH change as an indirect measure of rate of photosynthesis. A greater decrease in carbon dioxide concentration indicates a higher rate of photosynthesis. Okay, so as carbon dioxide is reducing, if there is a greater decrease in carbon dioxide concentration, it means the rate of photosynthesis is higher. For each bottle tested, the student added algal beads to a small bottle containing the indicator solution. That's the first step. The student is adding the beads to the bottle. Then they removed a sample of the indicator solution from the small bottle after some time. After some time, that means when the photosynthesis has occurred, they're going to take out this liquid there, this solution, from the bottle. And then they will use a colorimeter to measure the absorbance of the sample of the indicator solution. Though the student used a calibration curve to estimate the pH of the samples of the indicator solution. Suggest one reason why using algal beads instead of placing the algal cells directly into the indicator solution improves the validity of the investigation. We can easily separate the indicator. We can filter it out, that's why. Identify the two independent variables and the dependent variable in this investigation. So the independent variable is something that you are changing. So the first independent variable is obviously the wavelength of light, because you are putting different filters, so you can change the light. Therefore, we'll say wavelength or the color of light. Both of them are correct according to the marking scheme. Also, the other independent variable is the type of algae, the species of algae. That is independent because we are the one changing that. Now, dependent variable. So, how does these independence variable affect what is being measured when you are changing these variables? The absorbance of light. Colorimeter measures the it says here, colorimeter measures the absorbance of the sample. Okay, so we'll say absorbance or pH absorbance of solution. So we'll say absorbance of solution. You can also say the pH of solution. Now, in addition to the colored filters, algal beads, in addition to the colored filters, algal beads, small bottles, indicator solution, and a colorimeter, the student had access to standard laboratory equipment. The student carried out the investigation in a temperature-controlled room. Describe a method that the student could use to collect sufficient data so that the rates of photosynthesis of both species in the three colors of light can be compared. Okay, so you do have the independent and dependent variables mentioned. You need to describe a method to compare the rate of photosynthesis in both of these species of algae. The description of your method should set out in a logical way. That means you have to write steps or a paragraph that says what are these steps that we should follow, and be detailed enough for another person to follow. You should not include a risk assessment or details of how to prepare the algal beads. So this is for 8 marks. So this is basically a planning question. Let's see what exactly it is that we need to measure. Okay, so they're saying we need to measure the independent variable, or the variables that we are changing is, the first one is the color of the light. How are we going to change the color of the light? Using the filters as mentioned here. We have three filters for red, green, and the other one, red, green, and blue. So we can say we have a source of light. We can have a bulb here or a source of light. And then we will have a filter. We will have red filter, green filter, and blue filter. So we'll need to carry out experiment 3 times with each of these filters. And then what is the second independent variable? That is the species of algal beads. Okay, so we need to carry out the experiment with both species. Remember, there are 2 types of algae species. So we'll have the beads. The first set of beads will have one species of algae immobilized on them, and the second set of beads will have the other species of algae immobilized on the beads. So so for each of these filters, we need to carry out the experiment 2 times each with each species. Okay, and then what are we going to measure? We're going to measure the absorbance by the solution. How much wavelength of light is a solution, hydrogen carbonate solution, going to absorb? And that is going to be measured using a colorimeter. So we'll have our hydrogen carbonate indicator. Remember, when you this one at normal temperature, atmospheric concentration, it should be red. So we'll add our hydrogen concentrate solution and make sure that it is red in color, and also note down the have a known volume, so that when you're repeating the experiment, you use the same amount. This is a control. And then you will add your beads. Okay, the light will be turned on, light will fall on it, and after some time we'll see that the color has changed. We'll transfer this into a colorimeter and note down the absorbance. Let's write down the experiment now. So the first one would be, place a lamp and cover it with the red filter. At a known distance, place algal beads of one species. We will say place 15 or 10 algal balls, so that when you're repeating the experiment, you use the same number of beads. A known volume and concentration of hydrogen carbonate solution, which is red in color. Okay, before moving on, we can say carry out the experiment in a dark room so that the other wavelengths does not affect our experiment. After some time, we have to leave it for some time for the experiment to occur. Filter out the solution so that the algal beads and the hydrogen carbonate solution has separated. Then use a colorimeter to ensure that there is no zero error. First, we'll carry out the experiment with distilled water and make sure that the reading is 0. When you take a reading with distilled water, this is plain water, right? It has no color, so the colorimeter will show 0 reading. That's when you can ensure that when there is no color, the reading does show that there is 0 color. So now you can take your solution, which has changed its color from red to either magenta or purple or whatever, somewhere like that. It can measure that absorbance. Measure absorbance of solution using the colorimeter. For both species with these 3 different filters. So you have 6 experiments in total. 3 multiplied by 2. Okay, now you can repeat the experiment. Okay, so now you have obtained 6 readings for absorbance because you have carried out 1 time for each of the species with 1 color. Now you can repeat the experiment 3 times to get the most accurate results. For each color filter and species. Now when you are repeating 3 times for each of these, you have to find the mean. So you have to write and calculate the mean. If you need to get the mark. If you don't write the mean, calculate the mean, you will not get the mark, okay? So this is for 8 marks. We do have 8 points. We can add more points. For example, for a control experiment, carry out the experiment without the algal beads. Just use regular glass beads instead, and you will see that there is no Okay, I think that is going to be sufficient for this question. We have almost 10 steps. This is the 10th one points, and I hope this does help you. Let's move on to the next question. Identify a hazard in this investigation and state. Okay, so first you're supposed to identify a hazard and then state a risk associated with the hazard and state one precaution that the student should take. So first you need to identify a hazard, a danger, and a risk associated with that. How can it affect you? And precaution, how can you prevent that? So what's the hazard? There can be a lot of things. For example, you're using a hydrogen carbonate solution. It's a chemical. It can cause allergy, right? So the precaution would be use gloves or masks. You can also say you're using the algae beads. Some people might be allergic to it, or it can irritate them. Use gloves, masks, etc. Also, you're using the light source, and it can, if it's high voltage, it can there can be heat from the light source and it can cause burn. So do not touch the bulb or turn off the lamp before handling. So we can choose one of them. I will say the hydrogen carbonate solution. Now, for each color of light, the student carries out a statistical test to compare the pH of the samples taken from the small bottles that contain R saline and R costatum. The student decided that the t-test was the most appropriate test for these results. So just one reason why that a t-test was the most appropriate test for these data, because we're comparing the means of 2 sets of data, right? We're comparing the mean pH of the R salina and S costatum. So that's why t-test is the most appropriate. And then state a null hypothesis for this test. A null hypothesis basically means that there is no relationship or there is no difference between 2 variables that we are comparing. If you see any relationship that does exist, it's basically just a chance or error. We can say that there is no difference in the pH of samples, since we are comparing the pH of the samples in between both species of algae. You can say there is no difference in pH of samples between R salina and S costatum. Question number 2. A group of ecologists investigated the biodiversity of 2 peat bog ecosystems, A and B. Okay, so there are 2 ecosystems. One is A, the other one is B. What is a peat bog? These are basically wetland ecosystems that often contain rare species. Figure 2.1 shows an example of a peat bog. Now the ecologists sampled the plant species in peat bog A, and peat bog B. So the ecologists decided to use 2 suitable indices of biodiversity: Simpson's Index of Diversity and Shannon's Diversity Index. Okay, the ecologists had read that the 2 indices can lead to different conclusions. The Shannon Diversity Index, the second one, gives values in the range between 1.5 to 3.5, where 1.5 is low biodiversity and 3.5 is high biodiversity. Notice here that a lower number suggests low biodiversity, whereas a higher number suggests high biodiversity. Let's look at the table below. Okay, so this is table 2.1. You have peat bog A, one ecosystem, and then you have peat bog B. Both of them have different species, and the number of individuals per species is given in the second row for both of the peat bogs. Okay, so below that we have 2 index. We have Simpson Index as well as the Shannon index. Okay, so if you see for peat bog A, Simpson index has a smaller number, while peat bog B has a bigger number for Simpson index. If you look at the second row, Shannon index has a higher number now and a lower number in the second one. So the ecologists concluded that one index indicated that peat bog A had more biodiversity, which is this one because there is a bigger number in peat bog A, but the other index indicated that peat bog B had more biodiversity, which is Simpson index because Simpson index has a bigger number. So if you see, they're kind of conflicting. One index shows that peat bog A has more diversity, whereas the other index says that the peat bog B has more biodiversity. So just how the differences in the data in the two samples shown in table 2.1 may have led to different conclusions. Okay, so we have conflicting conclusions. They're asking how is the differences in data of two samples could have led to this. So if you look at the table, let's look at the table in detail. If you see in peat bog B, all of these are 0. So we basically only have 1, 2, 3, 4, 5, 6, 7, 8 kinds of species. We have a lower number of species in peat bog B and a higher number of species in peat bog A. Therefore, we can say Shannon index, peat bog A has a higher number of species and a higher Shannon index. So for Shannon index, the number of species is more important. Let's write it down. Okay, now let's look at the second difference. So what is the difference between A and B? If you look, these numbers are greater, right? The total number of individuals are greater. Let's find the sum of all the individuals in both of these. So if you find the total sum for peat bog B, we have 135 individuals, whereas for peat bog A, if you sum it, you get 99. Okay, therefore it's Simpson index is higher. So Simpson index depends more on the number of individuals. We can also say that peat bog B has a smaller range of species abundance, but you can simply write this as well, that there is a higher number of individuals in B. This question may seem really hard at first, but don't panic and try to understand what's the difference. So we just looked to answer this question, we first looked at the differences between the numbers given below, and we saw that one index has a greater number and the other index has a smaller number, and then on the other side the other index has a bigger number and the second index has a smaller number. So we can say that there's a difference because of that. And then we looked at the table. Can you see any big differences? We saw that these are 0. That means there's less number of species. This could be one difference. And the second one was we have, you can see that these numbers are a lot bigger than these. So the total number of individuals are more in peat bog B. You can also write down the number that you can see peat bog B had 135 individuals, where peat bog A only had 99. Okay, so and this way you can try to answer that question. Okay, moving on. The ecologists sampled the invertebrate species in peat bogs A and B. Okay, so they took the invertebrated species and they sampled it and calculated the invertebrate biodiversity using Simpson's index of diversity. The ecologists sampled invertebrate species in peat bog A between 13 and 16 o'clock on 3 different days within a 30-day period. Okay, so the first one says that within 30 days, on 3 different days, so could be after every 10 days. After every 10 days, they sampled it, and the time was always between 13 o'clock to 16 o'clock. Okay, next point is at 10 random sites along the edge of the path in the peat bog. So they took from that peat bog, they took 10 random sites along the edge of the path. By sampling the invertebrates within 1 meter square quadrat placed at 8. By sampling the invertebrates within 1 meter square quadrat placed at 8 sites. by using an identification key to identify the species, by counting the number of individuals of each species. Okay, describe how this sampling method could be improved to make sure that results are more representative of all the invertebrates in the whole of Peat Bog A. This is for 3 marks. So this question asks us, how can we improve our sampling method to make sure that all of the invertebrates are represented? Okay, so if you look at the first point, sample at different times of the day and at night. They're only sampling between 1 o'clock and 4 o'clock. Some invertebrates might come out at night or at different times of the day. So make sure that you're sampling at different times of the day and at night. Sample at different times of the year, not just within 30-day period, because it may change with seasons, right? Some invertebrates may come out in different seasons. And then samples at other sites, not just the path. You should not just sample along the path. Make sure that you're sampling in the entire peat bog at different sites, so you can actually make sure that the results are more representative of all the invertebrates. Some invertebrates might be more abundant in the center of the peat bog, for example. And then use an expert or identification app instead of just using this identification key to make sure that you are accurately identifying the species. You only need 3 points. You can choose any 3 of these. Now, table 2.2, this table shows the results of the invertebrate sampling in Peat Bog A. Okay, so after sampling it, they use this formula to find the Simpson index of diversity. Okay, so which is D is 1 minus sum of n over capital N squared. Now, small letter n represents the number of individuals of each species present in the sample. Remember here, these are the number of individuals, right? So that's what small letter n represents. What about capital letter N? It represents the total number of all individuals of all the species present in the sample. That is the total number, which would be 99 in this one and 135 there. But this is given here. So n is a number, for example, for the first species, the number of individuals present is 11. For the second one, it's 72. And so on. For n divided by n is 11 divided by total number of species, and we're getting this answer. And then n by n squared is this. So you're asked to use the formula, complete table 2.2 first, and then use the formula provided to calculate the Simpson's index of diversity. So you know small letter n and you know n by n, but you do not know capital N. To find out n over n, let's take this. So we can say n over n equals to 0.462. Now we know a small letter n is 72 divided by n equals to 0.462. So 72 divided by 0.462 equals to n, and that equals to, for the last one, n over capital N is going to be 13 divided by 155.8, and that is going to be 0.083. And then to find n by n square, you just have to square 0.083 and you will get 0.007. Now you're supposed to find the Simpson's index of diversity. Wait, but before that you need to find the sum. So add all of these and then you'll get the sum, which is going to be 0.270. The reason we're writing it as 3 significant figures or with 3 decimal places is because you can see all of these numbers have 3 decimal places. Okay, all of these have 3 decimal places. Always make sure to double check your calculation, especially when you're adding all these numbers, because there's a chance that you can miss a number or two. Okay, now let's find the Simpson's index of diversity. The formula is given right here. This is going to be D equals 1 minus sum of n over n squared, that is going to be 0.270. This one right here, 0.270, and the answer is 0.730. take care of the decimal places again. These are all with 3 decimal places. Now, last question. The ecologist calculated d value for a d value of 0.710 for peat bog B. Okay, so for peat bog A, we have 0.730, and for peat bog B, we have 0.710. So it says, or it concludes that A, it has more biodiversity. Use your value of D from B to this one to compare the biodiversity of peat bog A and peat bog B. We can say peat bog A, it has more biodiversity because it has a greater value for Simpson's index. Okay, and that's it.